I have chosen the article The Regulation of mRNA Translation During Mitosis to understand the mechanism of gene expression regulation that undergoes during this process. The source of my information comes from this article, which I previously mentioned, written by Tannenbaum et al. I will now quickly go over the key summary points that I will further discuss during my presentation that I noted in the article. I will go over what is mitosis, does regulation of gene expression come into play, levels of mRNA transcripts that are present throughout the cell cycle phases, one example which is the EMI1 deactivation, and the APC complex function. So mitosis is the process of cell replication that results in identical daughter cells. This is an extremely fast process which would not be possible without the regulation of gene expression of specific proteins. So that answers question number 2. mRNA transcript levels were measured during different points of the cell cycle, and many genes were either downregulated or upregulated, which further supported that gene regulation is essential for the progression through the cell cycle. So EMI1 mRNA must be deactivated in order to activate the anaphase promoting complex, or APC for short, and this deactivation occurs through none other than translational repression and protein degradation, further supporting the fact that we use gene expression regulation. And finally, the APC complex function in a nutshell will induce the progression and exit from mitosis. I will now be going over the major findings that I found in this article through a Jeopardy-styled question and answer. So we have different topics that are going to be presented, which include the background information, translational control, transcriptional control, key findings plus the support, and further studies. In mitosis, you have dividing cells that pass through multiple phases of the cell cycle. And in these phases, you have many proteins that are used to regulate each phase. As the levels of these proteins change, this is what causes them moving on to the next stage of the cycle. One of the major proteins that are used in the cell cycle includes the anaphase promoting complex, or APC protein for short. And basically, these are required to destroy other proteins at the end of mitosis in order to progress through the different phases. The takeaway message of the mRNA change is that they undergo gene-specific regulation that are translation repressed instead of being activated. EMI1 is one of the major genes in mitosis in which you have what happens is that EMI1 needs to be inactivated in mitosis so that the APC can activate and progress throughout the next phases of the cycle. EMI1 mRNA is suppressed during mitosis to keep the levels low in order for the APC to activate fully. The methods used to study the level of gene expression include RNA-seq and protein mass spectrometry. Next, I'm going to be talking about the translational control, which is one of the major examples of how the gene expression is regulated in mitosis. A major example of the translational regulation that occurs in mitosis has to do with the global translational repression of the bulk of mRNAs that are present in the different phases. So basically, what happens is that the translational efficiency will end up being very largely affected. And because of this, you have a large majority of the mRNAs translationally repressed at the mitotic entry and then translationally reactivated at the mitotic exit. Mitotic phosphatases are also regulated, and they are regulated in a downregulated fashion. Centrioles and histones are also regulated in a translational regulation mechanism. Phosphatases are strongly inhibited through phosphorylation and binding to inhibitory proteins. Mitotic translational repression represents an additional backup mechanism in order to inhibit protein function. So there are two key regulators for centriole duplication that occur. Histones show a strong reduction in protein synthesis in the M phase, and newly synthesized histones will not incorporate into condensed mitotic chromosomes, which explains why their translation is reduced during mitosis. So without translational regulation in mitosis and the cell cycle, you wouldn't be able to have progression through the entry or the exit phases, which means that you require this at the level of gene expression in order for mitosis to even occur as a whole. I'll go into some of the transcriptional controls that are underwent in the process of mitosis. transcriptional changes that occurred in mitosis weren't as varied as with translation. They affected G1 and G2, as well as they had gene-specific regulation in the M phase, where the mRNAs actually stayed constant. So the translational efficiency actually remained constant for items that were transcriptionally regulated, and these results basically indicate that gene-specific translational regulation affects a different set of genes. I will now go into the key findings plus the support for them. So mitotic translational repression is dominated predominantly by the global translational repression and the potent repression of the mRNAs. Main findings was that EMI1 mRNA is going to be repressed because this will enhance the post-translational protein inactivation, which overall causes APC to become activated and allow that further entry into the phases of the cell cycle. In mitosis, the speed of the translational and transcriptional regulation also plays an important role. Regulating transcription for altering the protein synthesis is very slow. This limits the usefulness of the transcriptional regulation to longer transitions during the cell cycle. Translational control alters protein synthesis almost instantaneously, which means it's well suited for mitosis. Translational control also can include rapid reversibility, which enables the protein synthesis to restart quickly as soon as cells will exit from mitosis and enter into G1. So the supporting evidence for the major finds of the transcriptional and translational regulations basically comes from ribosome profiling, which is measuring the ribosome occupancy of the native transcripts, as well as the use of a non-native reporter mRNA, which actually provides real-time and live readouts of the protein production that results from translation. Finally, I will be going into some further studies that also include the regulation of gene expression at the level of translation and transcription. Findings in this article was compared to mitosis in the somatic cells to the translational regulation during meiosis as well as early embryonic development. So basically what happens is that transcription is not initiated until the mid-blastula stage of development, and because of this, translational regulation is the main mechanism by which protein synthesis can occur. Translational regulation includes modulating the poly A tail length, which also occurs in mitosis but isn't a general mechanism for controlling the translational efficiency in somatic cells. So, as mentioned previously, mRNAs are usually translationally repressed in mitosis, whereas the mRNAs are usually translationally activated in meiosis. So all in all, the EMI1 gene is translationally regulated in both mitosis and meiosis, and this just overall goes to show that whatever the situation is, different processes, having translational regulation is very important at the level of gene expression in order to cause such vast differences in certain things such as daughter cells or unique cells.